Hello friends, this is Lieutenant Dr. Yasin from Abhidananda Senior College, Department of Chemistry. Today I'm going to discuss about the overview of a biochemical concept. As we have seen the before lecture that I have given you the introduction of the biochemical molecules, biomolecules. Now today we are going to discuss about the how we can overview the biochemical concept. So here we can have the molecules of life in the under the flag of the under the big flag of the biochemistry. How molecules of life. Many of the most important molecules in the your bodies are nothing but the polymers. You're going to be, shocking today because I'm going to give you a brilliant lecture about the biomolecules. Biomolecules of life is going to be happen. Proteins, carbohydrates, and nucleic acids are the extremely large molecules which is formed by the from smaller monomer subunits. Now you can see although the lipids are usually not considered to be a polymers, but the two are formed from the smaller molecules that have been linked together. So you need to extract relatively large amounts of proteins, carbohydrates, and lipids in your diet. Now complex reactions in your cells are use some of these molecules, and a few of them are they make a fourth group of the biomolecules, that is the nothing but the nucleic acid. The study of the chemistry of living things is called biochemistry, and the science explores the substance involved of the life process and the reaction they undergo. And the other than the water, which can account for 80% or more of them weight of an organism's, most of the molecules of the life, the biomolecules are organic in nature. A protein is an organic polymer which composed of amino acids which can bond it together in one or more chains. Now you can see an amino acid as a central carbon atom which can place into the center, which are bounded a carboxylic group, and amino group or hydrogen atom, and a variable side chain design as are shown in the structure. You can see the variable side chains are there. Amino group on the left hand side, in the carboxylic group on the other side, and these are the structure of amino acids. The C double bond O and OH is going to be having the variable side chain as well. Now, amino acids bonded to each other by forming a peptide bond. It's a very, very important bond, that is a peptide bond, and amide group form constant condensation reactions had taking place into the action, and then between the carboxylic acid group and the amino acid and the amino group of the another. Now you can see the structure of amino acids, and the two main amino acids linked to a peptide bond, that is a C double bond O, and an H is formed a dipeptide bond into the action, and this is the reaction is given to the in the screen, that is the C double bond O, and the NH, the bond is a peptide bond. Now you can see the chain of two or more amino acids which can link by peptide bonds is called as the peptide, and the term polypeptide is applied to a chain that 10 or more amino acids. The number of molecules will increase, then the peptide bonds are also the increase. In the proteins may have one or several polypeptide chains, and each chain must have an exact sequence of an amino acid. To the see these are proteins can fold. These are the structure of one. Proteins can fold into the either round or globular structure, which can or long fibrous structures, yeah, the hemoglobin, and you can see the collagen, and you can see the amino acid chains are held in place in three-dimensional structure by attractive the forces between the side chains of a different amino acids, which have been brought close together with the binding and the folding of the polypeptide chain. This is the very, very important terms about the proteins. I can see the enzymes. Many of the proteins in an organism act as enzymes. These proteins are catalyzed by chemically, which can spread the reaction up, and the allowing the reaction to take place at the lower temperature. Now you can see the substrate are brought close together. These are the what we call the enzymatic action. We are going to see here in the active sites of an enzyme, which lowers the activation energy of the reaction by facilitating the bonding together of the substrate to form a product. We can see the shade of the substrate is going to be form the complex, which can, and the active sites of the enzymes which can going to be unchanged to the product. Then you can have the very nice the enzymatic action. Now next phase is what the carbohydrates. It's an familiar carbohydrates includes the sugar, starch, and the cellulose as well. The carbohydrates contain carbon, hydrogen, and oxygen into the 1:2, 2:1 ratio. Now we can see the monosaccharides. These are the very, very important term, monosaccharides. The simplest carbohydrates are the simple sugars, or you can see the monosaccharides, which commonly have 5 or 6 carbon atoms to the action. Now you can see the glucose, the main ingredient of the corn syrup, and the is familiar to the monosaccharide. The glucose has the molecular formula that is a C6H12O6, and it can be represented as follows in the structure. You can see the cyclic form of the glucose and the open chain form of the glucose that has been rectified into the action. The most common simple sugars are glucose, fructose, and the ribose. The fructose has given to the form of the chain, and the ribose is also at the cyclic form. These are the structure we can have into the look into the on the screen. Now you can see the polysaccharides. After the monosaccharides, you can go for the polysaccharides. A polymer of a many monosaccharides bonded into the chain is called as the polysaccharides, and the starch is the polysaccharides that contains only of a glucose in it. You can see into the potato, the cut of the potato. You can see the place of the starch, and you can have the different structure of the glucose, which in form into the subunits. The plants also link glucose unites together in the cabbage. You can different way to form the polysaccharides is shown in the cabbage. You can say it's a cellulose which forms the plant cell as a wall. You can see the animal storage of the glucose as well as in the polysaccharides. You can call as the glycogen, which is similar to the starch, which is also having the glycogen and subunits of the glycogen. Now you can see the lipids. Lipids are the non-polar substances, which includes the fats, waxes, and oils produced by living organisms. And the lipids are not polymers. You can understand. You just remember this. These lipids are not polymers. These are their chemical structures vary widely. That's why it contains the carbon and hydrogen in a ratio of 1:2, with only a small amount of hydrogen. The fatty acid. The fatty acids. The most familiar lipids are the plant oils and the animal fats, which can be are the esters of the fatty acids, you can say, which are the carboxylic acid with a long and straight hydrocarbon chains, which usually having between the 12 to 24 carbon atoms. Now you can see the saturated the fatty acid. The simplest fatty acids are the saturated fatty acid, which have no double bonds between the carbon atoms, and the stearic acid is formed in the pork and the beef tissue. You can see the structure of the pork and you can see the beef tissues as well. Now you can see the monosaturated fatty acids. These are the fatty acids who have one or more double bonds between the carbon atoms and as a result as an unsaturated fatty acid. Oleic acid is a major component of the olive oil. You can see the structure of there. You can easily identify the structure of the monosaccharide. Now you can see the triglycerides. Animal fats and the plant oils are made up of primarily of triglycerides, molecules in which you can three fatty acids are bonded to a glycerol. You can see on this phase of the structure behind the screen. You can see the glycerol molecule by ester, which leakages as shown in the figure. You can try the three molecules of fatty acids are bonded together to form a glycerol, and you can see the other lipids. And under the class of lipids, the steroids consist of compounds those whose basic structure is very those of other lipids. As you can see here, the cholesterol, vitamin D, and some hormones are steroids. You can see the basic structure of the steroids. You can see and you can understand how the functions of the lipids are going to act. There are two major biochemical zones in the body. One is the excess energy absorbed by an organism can be stored as lipids, and this is why humans stored body fat if they eat too much or move too little. The second use is a chemical messenger, that is a hormone. Many steroids are hormones in the action. Nucleic acid, the sequence of the amino acids in the protein is determined by the genetic information coded into the long chain polymers, which can be called as the nucleic acid. The monomers that makes up nucleic acid are called nucleotides. Each nucleotide is made up of three parts of a phosphate group, that is a 5 carbon sugar, and a nitrogen containing cyclic compound is called as the nitrogen base. The base of the nitrogen is very important in this action. The structure of the nucleic acid nucleotide is see here, the phosphate sugar so group is showing into the action. the sugar is there, and the nitrogen containing base is also showing into the action. Now look at the phosphorus sitting in between, then the nitrogen is going to act with the carbon, and it's going to be having a alternate double bond, and as the not presence of the, these are the saturated things. Nucleic acid, which can have the phosphate, sugar, phosphate, these are the base, and these are the sequence of the nucleotides which can follow the phosphate, sugar, phosphate, sugar, and phosphate, sugar, which alternately we can have the bases. Now you can see that common nucleic acid are DNA, that is the deoxyribonucleic acid, and RNA, that is the ribonucleic acid. It contains DNA contains the sugar deoxyribose, and the RNA contains the sugar ribose. In DNA, the base thymine bonds with the adenine, and in RNA, thymine is replaced with the uracil. DNA is a double standard structure strand, and RNA is a single strand. That's why the structure of DNA, you can see this model of DNA, a portion of this molecule clearly shows its complexity between the molecules. You can see the single DNA molecule contains the many thousands of the nucleotides as well. Now you can see the base pairing of the molecules. The two chains of a DNA are held together because of the nitrogen bases, and of one chain are hydrogen bonded, and the other is a nitrogen base of the other chain. Now you can see the four different nitrogen bases are found in DNA, that is the adenine, guanine, you can see the cytosine, and the thymine as well. The adenine hydrogen bonds to thymine, and the guanine hydrogen bonds with the cytosine, and the order of these four pairing is nitrogen base is along with the one of the DNA chains provides the information for the sequences of the amino acids and the protein. Cell mechanisms read the DNA sequence, that is in groups of three bases called as the triplets, and the each triplet codes for a specific amino acid is or tells us the cells to start or stop making a protein as well. You can see the question number one: how label the amino group and carboxylic group of the dipeptide. You can see the answer as well. The amino group is going to be happen with the combination with the dipeptide, and it give you the attached with the carboxylic group as well. Question number two: draw an arrow pointing to the peptide bond. You can see the structure. You can easily draw. I give you the answer how you can going to be peptide bond with the dipeptide things. These are the answer for the questions. Now question number three: draw a square around each variable side chain. Now you can think about the side chain, how it's going to be act. Now you can see the answer. The R1 and R2 will be the different side chains, which is combination with the between N and H. That is the dipeptide bond is going to happen. Now you can see the what is the glucose is an polysaccharides, amino acid, or part of cellulose. You can see the answer is going to be happen with the what are the polysaccharides, amino acid, part of cellulose, 5 carbon sugar. This is the question for your assessment. This is the basic assessment question for you, what we're going to be understand. I think I have covered the all of the points here. Now thank you so much for the being patient listening to me. Thank you so much. We'll connect to the next video. Thank you so much.